going to introduce an extremely important part in our body, DNA, deoxyribose nucleic acid.
DNA is a type of nucleic acid, which is one of the organic compounds.
DNA is a long-chain molecules that encodes the genetic characteristics of a living organism. These instructions are needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to produce proteins.
Most DNA is located in the cell nucleus, and you can find some DNA in mitochondria. Also in most plants and animals, and DNA is packaged with proteins into a compact structure called chromosomes. And look at DNA closely, we can see the structure like this. And polynucleotide is a monomer of DNA, and which is composed of many nucleotides. One nucleotide in DNA is composed of 3 parts: a phosphate group, a deoxyribose, and which is a can of sugar, and one of the 4 nitrogenous bases, and which include adenine, cytosine, guanine, thymine.
The deoxyribose and phosphate group together is called the backbone. There are covalent bonds formed between phosphate of one nucleotide and the pentose sugar of the next nucleotide. This backbone ensures the store of genetic info in the bases is stable and secure. And the nitrogenous bases are divided in 2 parts, purines and pyrimides. Purines are larger bases composed of double rings of carbon and nitrogen atom. Adenine and guanine are included in this type. Pyrimides are smaller bases composed of a single ring of carbon and nitrogen atom. Thymine, uracil, and cytosine are included. 3 hydrogen bonds formed between adenine and thymine, while 2 hydrogen bonds formed between cytosine and guanine. A female chemist named Rosalind Franklin contributed to the structure of DNA using her X-ray diffraction pictures. Crick and Watson then used this evidence to test and build the model of DNA. The DNA is a double helix made of 2 antiparallel strands of nucleotides linked by hydrogen bonds between complementary bases. Each strand consists of a chain of nucleotides linked together by covalent bond. Just like what we've discussed previously, hydrogen bonds are formed between bases, and adenine is always paired with thymine, guanine with cytosine. These bases complement each other. This is what we call complementary base pairing. The phosphate is linked to carbon-3 of the deoxyribose sugar, which is referred to 3'. The deoxyribose of another nucleotide is linked to the phosphate to the carbon-5 5' terminal. One strand is oriented in 5' to 3', while the other strand is oriented in 3' to 5'. 2 strands run in opposite direction. 2 strands are wound together to form the double helix. So, how organisms replicate their DNA? Scientists came up with 3 basic models. Conservative replication, which is one molecule that consists of both original DNA strand and another molecule that consists of 2 new strands with exactly the same sequences as the original molecule. Semi-conservative replication, 2 DNA molecules with 1 original strand and 1 new strand. And dispersive replication, 2 DNA molecules that are mixtures each individual strand. Mendelssohn and Stahl conducted their famous experiments on DNA replication using E. coli bacteria. Stage 1. Began by growing E. coli in N15 medium, which is a heavy isotope of nitrogen for generation. Stage 2. All DNA is of a heavy type containing N15 and the sample of E. coli is saved. Stage 3. E. coli are transferred to N14 medium, a light isotope of nitrogen. All new DNA will be synthesized with this isotope. Stage 4. After the E. coli have divided a second time, DNA is extracted from all samples and added to calcium chloride, then put into the centrifuge. Densities of the DNA are measured using density gradient centrifugation. This method separates molecules such as DNA into bands by spinning them at high speeds that forms a density gradient from top to the bottom of the spinning tube.
The DNA of the cells grown in N15 medium had a higher density than cells grown in normal N14 medium. After one division, DNA molecules were found to contain a mix of N15 and N14, disproving the conservative model, because the conservative model would have predicted two distinct bands in this generation: a band for the heavy original molecule and a band for the light, newly made molecule. After two divisions, the pattern of two distinct bands is shown, one at the position of a mixture molecule containing N15 and N14, and one at the position of a light molecule consisting solely of N14. And this disproved the dispersive model, because in dispersive replication, all the molecules should have bits of old and new DNA, making it impossible to get a purely light molecule. Helicase is an enzyme responsible to unwind the DNA molecule, and it will begin the process at the origin of replication. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y shape, known as the replication fork. This area will be the template for replication to begin. Once the DNA is unzipped, single-strand binding protein will come and keep the strands apart. Primase is a type of RNA polymerase that generates RNA primers. Primers are short RNA molecules that act as templates for the starting point of DNA replication.
Enzymes known as DNA polymerase are responsible carrying the new strand.
DNA polymerase 3 only add a nucleotide to the 3' end of pre-existing strand. 3 nucleotides are available in the area. One strand is oriented in the 3' to 5' direction, leading strand. Lagging strand begins replication by binding with multiple primers. DNA polymerase then adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single unified strand.
In the end, replication produced 2 DNA molecules, each with 1 strand from the parent molecules and 1 new strand.
The polymerase chain reaction is an artificial method of replicating DNA under lab, so that many copies of a selected DNA sequence can be made. PCR relies on a thermostable DNA polymerase, Taq polymerase. Denaturation. DNA sample is heated to 95 degrees Celsius to separate the two strands. Annealing. DNA sample is then cooled down to 55 degrees Celsius so that primers can bind to their complementary sequences on the single-stranded template DNA. Extension. The DNA sample is then heated to 72 degrees Celsius, the optimum temperature for heat-tolerant Taq polymerase to function. The number of DNA molecules can be doubled in each round of cycling, showing a pattern of exponential growth.
Okay, now let's move on to transcriptions and translation. Firstly, we need to get familiar with RNA structure. RNA stands for ribonucleic acid. RNA contains the 5-carbon sugar called ribose instead of deoxyribose in DNA. RNA includes the nucleotide base uracil instead of thymine. RNA is single-stranded.
Transcription is the synthesis of mRNA copied from the DNA-based sequences by RNA polymerase.
mRNA is an intermediary molecule that carries the message of DNA and the code to the cytoplasm. An enzyme called RNA polymerase binds to a specific site on the DNA and separates the DNA into a single strand. One of the strands will serve as template strands, or we call it anti-site strands, which has a complementary base sequence to the synthesized RNA, And the side strand, on the other hand, is the DNA strand with the same base sequence with the RNA.
RNA polymerase reads from 3' to 5' and generating mRNA from a 5' end. As RNA polymerase moves along the strands of DNA, acting as the template, RNA nucleotide floats into place by complementary base pairing. Uracil will be paired with adenine. RNA polymerase forms covalent bonds between the RNA nucleotide.
Transcription stops at the end of gene and completed DNA molecule is released. RNA polymerase zipped DNA back as it goes, and RNA polymerase detached and DNA double helix reformed.
Genetic code enables the conversion from the base sequence on RNA into an amino acid. Living organisms use triplet code, this group of 3 bases called a codon. So after mRNA is released into the cytoplasm, it binds to a ribosomes, which consists of large and small subunits with ribosomal RNA within. Ribosomal RNA, which is known as rRNA, is a composite of each ribosome. Transfer RNA, which is known as tRNA, is a RNA that helps to decode and to translate the message within the nucleotide sequence of mRNA into specific amino acid sequences.
Ribosome bind to mRNA at a start codon AUG and moving along the molecule in a 5' to 3' direction. Each tRNA has an anticodon, a set of 3 nucleotides that bind to matching mRNA codon through base pairing, and the other end of the tRNA carries the amino acid that's specified by the codon. As tRNA enter the ribosome and bind to the codon, the amino acids are linked to a growing polypeptide chain bonded with a peptide bond.
The ribosome moves along the mRNA molecule, synthesizing a polypeptide chain until it reaches a stop codon and the polypeptide chain is released.